Okay, let's dive into something really fascinating today. Ever wonder if the camera in your pocket, or maybe just regular consumer DSLR, could actually be, a serious scientific tool? Our deep dive is all about that. How researchers, students, even turfgrass managers can use traditional cameras to measure plant health. Pretty accurately, too.
Yeah, it's quite surprising what you can get out of them.
We'll look at the insights these images give us and, importantly, how things like image formats and digital corrections affect the data you get.
Right, those details really matter.
Think of this as your quick guide to a field that's cutting edge, but also, well, surprisingly easy to get into.
Exactly. The accessibility is a huge part of it. We're talking about using, really affordable cameras. The study we're looking at used a Nikon DSLR from 2013.
Wow, okay, so not exactly new tech.
Not at all. And it lets people do quite advanced plant phenotyping, measuring plant characteristics without breaking the bank. It's a solid alternative to counting leaves or rating by eye.
Which takes forever and can be pretty subjective.
Totally. So this is great for smaller experiments, maybe for students or for commercial managers keeping an eye on things alongside professional researchers, of course.
So making science more accessible. I like that. How did they actually set up the study to, prove this worked for something like turfgrass?
Well, the setup was actually pretty straightforward but effective. They took pictures of TifTuf Bermudagrass, that's a common type you see on golf courses and lawns.
Okay.
Grew it in simple PVC pipes, basically acting as lysimeters to control water. And they put these inside custom-made light boxes.
Light boxes, why?
To keep the lighting conditions consistent. They were inexpensive too, just built for the job. Then they messed with the conditions, different mowing heights, different watering levels.
Mimicking real-world stresses.
Exactly. And they kept track of all the environmental conditions in the greenhouse, temperature, humidity, all that, for context.
Okay, so they got the pictures.
Right.
Then what? How do you turn a photo into, hard data? That seems like the tricky part.
Right. That's where the software comes in. They used open source tools, Python scripts, things maybe like Turf Analyzer, that kind of software.
Okay.
To analyze the images for different metrics of plant health. And honestly, they found some correlations that were, pretty surprising.
Oh, yeah. Like what jumped out from just looking at a standard photo?
Okay, get this. The percentage of yellow turfgrass in the picture. They called it yellow.
Not green?
Yeah, yellow. And this simple number, pulled from a regular RGB photo, matched up really closely with human visual quality ratings. The correlation R was minus 0.89, super strong inverse relationship.
Minus 0.89. Wow. So the more yellow, the worse the visual rating. That makes sense.
It does, but it proves that this yellow measure isn't just fluff. It's a really solid indicator of what people see as quality issues, maybe even spots problems earlier.
That is a big deal.
Yeah.
Okay, so a normal camera picks up on quality through yellow. But what about greenness? The thing we usually focus on for healthy plants.
Yep, they looked at that too. The percentage of green turfgrass per G also pulled from the same photos. Yeah. It showed a really high correlation with NDVI, that's the Normalized Difference Vegetation Index. The R value was 0.91.
NDVI?
Yeah.
Isn't that something you usually need special expensive sensors for? Multispectral cameras.
Often, yes. NDVI is a standard measure for green biomass, for plant figure. So finding that per G from a cheap camera tracks it so closely, that's huge. It means you might get similar insights without the costly gear.
Okay, this is getting really interesting. Did they push it further? What about even more advanced measurements, things beyond basic greenness?
They did. They calculated some new metrics right from the color camera data. For instance, something called the hue difference illumination ratio, or HSVI.
HSVI.
That correlated strongly with near-infrared reflectance, NIR, R value of 0.891.
And NIR is important because
healthy plants reflect a lot of NIR light. Stress plants reflect less. So HSVI could be a proxy for that stress indicator.
Got it. Any others?
Another new one, the C-Lab B to a ratio. They just called it BA. It's basically a measure of the color balance, yellow-blue versus red-green.
Right.
And that correlated pretty well with red edge reflectance, RE, at 0.734, and also NDRE at 0.902.
Red edge and NDRE, those are key for spotting stress even earlier, right?
Yeah.
And nitrogen levels.
Yeah.
Often needing even more specialized cameras.
Exactly. Things that usually require hyperspectral or very specific multispectral sensors. So the potential to estimate these from standard photos is, it could really open things up for wider use.
Okay, and beyond single metrics, they combine some. there was the COMB2 index.
Yeah, COMB2. It was a more complex index using several of these photo-derived metrics, and kind of surprisingly, it had its strongest correlation with water use.
Water use, really?
Yeah, R equals minus 0.60. Now, that's not quite strong enough maybe to predict exact water use on its own, but it definitely picked up differences between the experimental treatments, like the different irrigation levels.
Interesting. So it hints that these combined metrics might tap into really complex plant responses.
That's the idea. Still needs more work, but promising.
Okay, so we're getting all this amazing data from basic cameras. But you mentioned formats and corrections earlier. This seems critical. How much does processing the image actually change the results? Do you need to do all that stuff?
Yeah, the million-dollar question. It's nuanced. The study looked at standard JPEGs versus TIFF files derived from the camera's raw data.
Okay, JPEG versus TIFF RAW.
And corrections.
And corrections like color correction CC, trying to make the colors more accurate, and geometric lens distortion correction LC, fixing warping from the lens.
So what did they find? Did applying all these fixes make the data better?
It was mixed, honestly.
Yeah.
Sometimes, yes, the corrections slightly improved how well the photometrics matched up with the reference measurements, like the NDVI or visual scores.
Slightly, like how much?
Often pretty minor. For most metrics, the R-squared value, which tells you how much variance is explained, increased by less than 2%. That's not a huge jump.
Right, 2% is pretty small. But for that COMB2 index we just talked about, the one linked to water use, applying the corrections made a big difference. The R-squared value jumped by 0.310.
Whoa, 0.31. That's substantial. So corrections really mattered there, but not so much for the simpler greenness or yellowness.
Exactly. And that's the key takeaway here. Slapping on every possible correction isn't always needed, and sometimes it might not even be helpful.
How so? Could it make things worse?
It could. It adds complexity to your process, for one, and sometimes corrections can introduce their own small errors or artifacts. Think about over-sharpening a photo. Sometimes it just looks worse, more noisy.
Okay, that makes sense. So you need to be careful.
Definitely. The decision to use corrections and which ones really depends on what you're trying to measure. We probably need clearer standards or guidelines on when these adjustments are truly beneficial and when they might be overkill.
So wrapping this up, it seems clear that, yeah, affordable cameras are surprisingly powerful for this kind of plant analysis, like really powerful.
Absolutely. Democratizing the science, like we said.
But the deeper insight, maybe, is understanding how the image itself, the format, the processing, can change the story the data tells you. Sometimes subtly, sometimes quite dramatically, like with that COMB2 index.
Right. It's about being an informed user of the technology, knowing the potential and the pitfalls.
It's about being informed, not getting overwhelmed by the options. Knowing when the simple approach is fine and when you need that extra processing step.
Precisely.
So, maybe the final thought for you listening is this. What important information might be hiding in plain sight in your digital photos? Just waiting for the right analysis? And maybe a careful thought about how those images were created and handled. What details are you potentially missing or maybe misinterpreting?